Good day, everyone. Happy Wednesday, and welcome to our webcast. It's time to go ahead and get started. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix. It's my pleasure to introduce Dr. Sergey Kornilov of the Child Studies Center at Yale University's School of Medicine. Sergey was our first prize winner in our second annual abstract competition this past winter, and he is going to present his work to us today. With that, Sergey, I'll pass the mic to you.
Thank you very much, Cheryl. Good morning, everyone. I'm going to talk about some of our research surrounding a unique, geographically isolated population that we sampled from to study genetic basis of developmental language disorder, and I'm going to talk about some of our findings. So language disorders are defined as difficulties acquiring and using one's native language in the absence of other factors that could explain language disorders. So, for example, intellectual disability, sensory problems like hearing loss, severe psychiatric conditions, or known genomic disorders. It's a complex and common disorder. It affects approximately 7 to 10 preschoolers. In about half of the cases, it persists into adolescence and adulthood. And in adolescence and adulthood, it is associated with negative outcomes in a variety of developmental and functional domains. So, for example, it affects the quality of friendships and emotional attachments and personal relationships of individuals with a history of developmental language disorders. It also is associated with behavioral problems, oppositional defiance disorder, and substance abuse. It increases one's risk with respect to academic difficulties, obviously, as approximately half of the children affected with developmental disorders of language in the spoken domain also have problems reading, spelling, and so can be classified as dyslexic. And these traits and disorders are highly familial and heritable, with the average heritability estimates around 50 to 70%. They range wildly for different phenotypes. They tend to be higher for endophenotypes, such as phonological working memory measures that were proposed as both a clinical marker and endophenotype for developmental language disorder. And despite being one of the sort of most common neurodevelopmental disorders, DLD, or developmental language disorder, is largely understudied with respect to its neurobiological and genetic etiology. And I'm going to talk a little bit about the existing research on the topic and then transition to our studies. So what is interesting is that despite what I just said, despite developmental language disorders being sort of the least, one of the least studied classes of neurodevelopmental disorders, we actually, the field actually has a success story. In the late 90s and early 2000s, there was a very prominent publication by Simon Fisher and his group who identified a single point mutation in FOXP2 as a cause of a rare case of autosomal dominant orofacial dyspraxia, so a condition that is characterized by pronounced problems in comprehending and producing speech and language. In a unique three-generational pedigree in the United Kingdom. And so in this particular family, this single point mutation effectively resulted in only half of the functional FOXP2 protein available to members who were of the family who were carrying the mutation. And FOXP2 is a transcription factor. It actually regulates over 300 genes. It's highly expressed in the brain, especially in the areas that seem to be involved in different forms of skill learning. And it's associated with skill learning not only in humans, but also in sunbirds, and it's associated with some of the vocalization patterns in other model animals as well. And of course, members, in the bottom right here, you can see that there are some structural differences, bilateral structural differences in gray matter volume between the affected and unaffected members in this family. And despite sort of being one of the most famous cases of an identifying gene for a complex disorder, FOXP2 seems to be not associated with common forms of speech and language disorders, so developmental language disorders that are not monogenic but complex and common. So studies of FOXP2 led to the discovery of the role of CATNAP2, contact-associated protein 2, as one of the important players in neuronal development. So CATNAP2 is downregulated by FOXP2, and it's a member of the neuroxin family. It's a neuronal transmembrane. It codes for a neuronal transmembrane protein, and it's very important for the regulation of synapse formation and overall neural development, including such processes critical for cognitive development as axonal differentiation. And what is interesting is that common variants in CATNAP2 seem to be associated with antiphenotypes for developmental language disorders, so again, phonological working memory measures, and language measures in the specific language impairment consortium cohort, but also with early language development as assessed by parents in the general population, and also in general population of adults. It's associated with various neural indices of language processing, and those are the results of both EEG, ERP, and fMRI studies. What is also interesting about CATNAP2 is that, like FOXP2, it seems to be involved in processes that support vocal learning, primarily in songbirds, but it is also associated with a host of neurodevelopmental disorders beyond developmental language disorders, so with autism spectrum disorders, Tourette syndrome, schizophrenia, different types of epilepsy, and intellectual disability, which led some of the researchers in the field sort of label this phenomenon of the growing network of CATNAP2 associations as the erosion of phenotypic specificity, although undoubtedly CATNAP2 is one of the key players in neuronal development, but surely it's not the only not the only gene that regulates the development of neural systems that support the development and functioning of language. Other candidate genes have been located and identified through targeted associations in the regions that have been shown to be linked to speech and language difficulties in the UK specific language impairment consortium cohort. So, for example, Diane Newbury and her colleagues identified CMIP and ATP2C2 as two of the genes that are located in regions, in the SLI1 region on chromosome 16q, as associated with phonological working memory, as I mentioned, sort of a very prominently studied phenotype for developmental language disorder. And both of these genes seem to be involved in some sort of regulation of neuronal function. So CMIP is involved in cytoskeletal remodeling, and correspondingly in neuronal migration and formation of synapses, while ATP2C2 regulates the calcium homeostasis, which is, as we know, is also critical for neural function. And other genes have been identified. So our lab, for example, has contributed to the field by identifying a structural variant, a valence translocation that affected the SEMA6D gene located in chromosome 15q21 in a patient that had a pronounced developmental language disorder phenotype, sort of consistent with the general phenotypic presentation of the common forms of DLD, and the gene affected by that mutation is part of the plexine and semaphorin integrin signaling pathway. So it's involved in exon guidance, and there's a growing literature that is implicating semaphorins and integrins in early stages of neuronal differentiation. What is interesting, however, is that until very recently, our field has been in a sort of pre-GWAS era with several targeted association studies, but essentially no genome-wide association studies. That changed in 2013 when there was a set of studies published that utilized data from several large-scale cohorts. So, for example, Eicher and his colleagues performed a GWAS of comorbid spoken and written developmental language disorders using the ALSPAC cohort, and found an association with Zinsenberg 3-5D that has also been associated with sort of overall gray matter volume in the pediatric neuroimaging in cohort. With the exception of that, the only other set of findings that survived corrections for multiple testing, including these GWASs, are in the recent study by Simpson and colleagues, who identified several of the genes, including RKN3, have been associated with spelling and reading measures. Now you will notice that for those GWAS, the association has been found for spelling and reading rather than spoken language measures. And although this is great, those are not essentially language phenotypes per se. And it's a very complex landscape of findings. In general, if you look at the associations that did not survive corrections for multiple testing, but were nevertheless nominally implicated in these traits and these disorders in the published GWASs. And part of the reason why the patterns of findings so complex and I don't want to say unreliable, I want to say sort of unstable and tangible, but with a certain degree of difficulty, is there are several reasons for that. So first, developmental language disorders are diagnosed using a set of exclusionary criteria typically, right? So as I mentioned, it's a disorder where language is affected, but in the absence of sort of deficits in other developmental and cognitive domains. And this leads to the extreme heterogeneity of developmental language disorder in sort of clinically referred, for example, samples. And of course, it's complicated in part because of the same reason that others are struggling in the field of complex traits, right? It's the sort of well-known and hypothesized locus and allelic heterogeneity of common complex traits and disorders. But also, developmental language disorder is an especially sort of difficult trait to study, in part because language is such a multivariate trait, and different domains of language can be affected in different children, and that's sort of adds this additional layer of complexity to such studies. And finally, I just wanted to mention that Dorothy Bishop published this beautiful paper in PLOS in 2010, where she showed that actually part of the reason why we're sort of struggling in identifying the neurobiological basis of DLB is due to essentially lack of funding. So when you compare, this is total expenditure in thousands of dollars per year for studying all of these neurodevelopmental disorders, and actually specific language impairment, in Dorothy Bishop's opinion and mine, is sort of an important enough disorder to receive as much attention as ADHD or autism, yet it hasn't so far. Now, in order to sort of address some of the issues related to heterogeneity that I just mentioned, we performed a genetic study of the molecular basis of developmental language disorder in a unique Russian-speaking population. So we published before in this population, and initially it was referred to us as having sort of an unusually high suspected prevalence of language disorders among children. An epidemiological study that we conducted and published in 2013 suggested that over 30% of individuals in this population are affected by developmental language disorder, and that is not actually in comparison. You know, the graph on in the top right corner shows the prevalence of overall developmental language disorder in individuals of various age groups: preschool children, primary school children, excuse me, adolescents and adults, in comparison to another population that is located sort of in a rural region and is matched to a set of socioeconomic indicators to the AS population. Now, AS population itself has been geographically and culturally isolated for a number of reasons, and it was established in the early 15th century by several nuclear families, some, if not most of which, are suspected to have had individuals with developmental language disorders. Now, currently, this population includes approximately 860 residents, with approximately 120 children in between the ages of 3 and 18, and 82% all of the current residents are related through a complex 11-generational pedigree. Now, this is not to say that this population is inbred, actually, because in part due to religious reasons, close intermarriages are prohibited, and essentially these connections between individuals are relatively distant and indirect. And we suspected that this population, like other genetic isolates, would show a reduced allelic variability with respect to the etiology of developmental language disorder. And also, what was important for us is that in this population, the overall environment, socioeconomic status, income, sort of levels of poverty, everything is relatively uniform. So children go to the same schools and the same preschools. Actually, there's only 2 schools in the region, and there's very little, if any, variation in the socioeconomic status, familial education, parental education, and parental income. Now, in this population, nonverbal intelligence seems to be slightly lower than in the general population, right? So in the around 95 or something like that is the mean estimate. This is typical for studies of neurodevelopmental disorders and neurodevelopmental disorders that affect speech and language. In general, what is important is that it's the distribution of the IQ values in this population does not sort of suggest that it's generally characterized by sort of deficits in nonverbal cognitive functioning. The individuals in this population also do not seem to have increased rates of sensory problems and hearing loss or other neurological or psychiatric problems. Yet our studies showed that the deficits are very pronounced in the domains of expressive language, in the domain of receptive language, written language, reading and spelling, lexical semantic development, and development of vocabulary, and morphosyntactic development, and the ability to efficiently use grammatical information online, phonological development, and related traits, for example, theory of mind that has hypothesized to involve a strong sort of language component due to the dependence of this ability on the development of the ability to represent embedded grammatical structures. Right, and we also showed recently that we see evidence for deficits in linguistic as well as auditory processing in this population when we use neurophysiological measures using high-density electroencephalography and event-related potentials. In this population, the phenotype does not breed true, and actually, I'm not going to focus too much on this example or pedigree, but I'm just going to say that it seems that this, the complex phenotype travels in these sort of patches, skips generations, and in general, our segregation analysis suggested that the transmission of the phenotype in the population is not Mendelian. And in addition to, perhaps surprisingly, in addition to that, we found that sort of all of the possible combinations of affected language development domains are found in S with a certain frequency. And if you look at the overall population, and you look not only sort of as individuals who we classify as severely affected because they have deficits in at least 2 of the domains that we looked at, but if you look at all of the domains, you'll find that only 34% individuals in this population are completely unaffected. So, right, so over 65% have one impairment in one of the language domains. And we performed a genome-wide association study of developmental language disorder in this population using Illumina's 370K duo bit chip panel using a multivariate set of phenotypes. And the phenotypes for this study were derived from sort of ecologically sound measures of language development based on elicited speech samples. So narratives that children generated using wordless storybooks and or extended detailed interviews with the adults. And we looked at, of course, at associations with single nucleotide polymorphisms, SNPs present in the Illumina's platform, but also on other types of genetic variations, so inferred couple number variants and rare genetic variants that we studied in a sample of 12 severely affected proband using whole exome DNA sequencing. And the goal of this was the genetics, the first genetic association part, and then I'm going to talk first about that, and then I'm going to talk briefly about another recent study of ours that examined one of the candidates revealed in the association study with respect to its potential role in the development of neural systems that support language using the network connectomics approach. So endophenotypes derived using measures of electrical brain activity during language processing. Okay, so this is actually a relatively small genome-wide association study. You have to remember, though, that 359 individuals that ended up in the final analysis sample represent approximately half of the currently living population in AS. And we looked at, as I mentioned, at the associations between individual SNPs as well as performing gene-based association testing, CNV association testing, and examination of runs of homozygosity in this sample of 359 individuals using multivariate phenotypes that included measures of linguistic errors as well as measures of syntactic complexity, so the complexity of the output that children and adults produced when these samples were elicited. Now, perhaps not surprisingly, and sort of in line with what I talked about when we saw previously was findings for developmental language disorders, despite there being sort of a very interesting set of nominally significant individual SNP findings, none of those survived corrections for multiple testing individually, although you will see that for the syntactic complexity, there's a cloud of SNPs on chromosome 9q, and also you can see it very well, but there's a lot of SNPs on chromosome 18q that ended up in our top SNP list. And this is important, I'm going to talk about briefly about the two genes that were implicated in DOD using this GWAS. What I also would like to highlight is that the associations that sort of the most intriguing associations we found were only part of the DOD phenotype in the population, whereas the sort of the linguistic errors phenotype composed of measures of errors in use of lexical, grammatical, domains. So our associations sort of are limited to this part of the phenotype related to the syntactic complexity. Remember, I mentioned several SNPs on chromosome 9. Actually, if you pull together data from these SNPs, you will get a significant association of syntactic complexity and the TNC gene, and TNC codes for extracellular matrix protein tennessine C that is involved in neural development. It has recently been implicated in autosomal dominant deafness. It seems to play an important role in cochlear development, but also animal studies suggested that TNC knockouts show a host of structural and functional cortical development abnormalities. So, for example, knockout mice have much higher density of neuron in the cortical plate, but also show deficits in and atypicalities in dendrite morphogenesis and other sort of indices of the optimality of neural development. So that's one of the findings that we were intrigued by. But perhaps most importantly, we did find an association that survives corrections from multiple testing at the gene level. Right? So when performing a SET-based test, we found that SETBP1, which I gloriously misspelled on this slide, is significantly associated with syntactic complexity, as I mentioned, surviving corrections from multiple testing. So here you can see that there's a a bunch of single nucleotide polymorphisms in SETBP1 that seem to be associated with the phenotype in this regional association plot. And although very little is known about the function of the protein that SETBP1 codes for, so it codes for SET-binding protein that binds the nuclear oncogene involved in DNA replication, apoptosis, and transcription. And it has been shown to inhibit proteins that play important roles in regulation of the development of synapses and synaptic plasticity. Mutations in this gene cause so-called midface retraction syndrome or Schinzel-Gideon syndrome that is also characterized by pretty severe developmental delays, including delays in the development of language, but also sort of general cognition as well. And several independent reports in the recent years suggest that having one broken copy of the SETBP1, so having half of the functional protein, is associated with a phenotype that is distinct from Schinzel-Gideon syndrome. So in the bottom right corner, you can see a plot from Filges and colleagues, and it shows a hemizygous deletion of part of the SETBP1 in the patients with severe developmental language, expressive developmental language disorder. So I'm going to sort of come back to the potential role of SETBP1 in a few minutes, but before I do, I would just like to briefly mention our whole exome sequencing findings. And so we performed exome sequencing in 12 severely affected DLB probes from the AS population using Illumina's HiSeq 2500 platform. And we focused on variants that were present in at least 4 out of 12 individuals in our sample, were not present in any of the individuals in the control exome sample from the same region that we had, which was approximately 10 individuals, and also on variants that were not found in existing public databases at frequency higher than 5%. We prioritized these variants using ecstasy that sort of attempts to integrate data regarding the possible pathogenicity of the variants, haploinsufficiency predictions, and similarity to other candidate genes identified to sort of seed human phenotype ontology terms. And what we found is that there were 14 coding sequence variants carried by at least, so each one of those was carried by at least 4 out of 12 severe DLD programs in the population. And all of these variants affected genes that regulate neural development in one way or another. So we found friendship mutations in NT5 DC2, missense single nucleotide variants in NECAB1 and ILK, and missense variant in cadherin2 gene that is actually a known mutation that is carried by less than 2% of the general population, but it's actually present in 7 out of 12 individuals in AS. And all of the findings, for example, on this slide, are predicted to be pathogenic. What I'm not going to talk about is but what I would like to mention just briefly is that there is actually seems to be a sort of a common pathway for a lot of these genes that are regulated by a set of common transcription factors, and this is all I'm sort of prepared to say about that pending this publication. But now going back to our main findings at BP1, we performed, as I mentioned, another study that looked at the role of SETBP1 in neural development and in sort of the efficiency of linguistic processing in the AS population. And we, as I mentioned before, we performed several EEG and ERP studies in this population for the goal of illuminating sort of the neurocognitive architecture of the disorder. But we also decided to focus on connectivity patterns in the study, in part because there is a host of recent publications that implicate atypicalities in connectivity patterns. So, for example, overconnectivity and underconnectivity. Both have been implicated in autism spectrum disorders, but also attention deficit hyperactivity disorder, intellectual disability, and so on. And these patterns seem to be under very strong, probably not the same very strong, but under strong genetic control, with they're approximately 23 to 89% heritable, with a median estimate around 65% as far as I remember. And we examined the role of SETBP1 in DLDNS by performing a targeted association study. So we looked at 5 SNPs located in SETBP1 in the mixed sample of affected and unaffected children from the population, using electroencephalography and event-related potential indices of neuronal functioning during word processing. And what we did is we essentially obtained measures of connectivity between 18 regions of interest that seem to play important roles in language function in general, based on the sort of several high integrated reviews of the neurobiological foundations of language. And we converted these connectivity measures into graph networks and used higher order properties of these networks. So, for example, how cohesive is the resulting functional network as endophenotypes for the association analysis. And so in this electrophysiological experiment, what children did is they sat in front of the screen. They were wearing an electrode cap with 64 electrodes that we were measuring the electric activity of their brain at the scalp during word processing. So children were sitting in front of a computer, looking at the screen. There was a picture, and then they heard a word, and the word either matched or mismatched the picture in several ways. I'm not going to go into detail sort of regarding the findings of that study, except for sort of in the bottom right corner, you can see that a mismatched word produces a strong so-called N400 component in typically developing children in the population, and a much smaller N400 in children with developmental language disorder in the same population. So we analyzed these data using measures of lacked functional coherence between 18 different bilateral language regions of interest for language in the intercortical space, using low-resolution tomographic estimation of and source localization in 6 different EEG bands. So alpha, theta, delta, and 3 betas, corrected the results I'm going to be talking about are corrected for multiple comparisons. So what we found is that one of the main focuses of our study, RS-808-5464, which is one of the strongest associations in the GWAS study that I mentioned, explained 26% of variance in the cohesion of the network, of the language network during word processing in the alpha EEG band. And essentially, having an extra copy of a minor allele increased the coherence by 3/4 of the standard deviation. For some reason, do not see the JERP score here, but this is a very highly conserved region. And what is also interesting is that this increased cohesion seems to be associated with depressed language development. So having a risk allele at RS-808-5464 not only increases the cohesion of the intercortical language network, but it also leads to depression in the language scores. And this seems to be associated with a pattern of overconnectivity that we see in this population that is associated with common variants in SETBP1. So in the alpha EEG band, there's at least 11 pairwise coherences that we found were significantly stronger in carriers of the minor allele at RS-808-5464 compared to individuals who were not carrying minor allele. And the majority of these sort of increased functional connections are interhemispheric rather than intrahemispheric, and they implicate left auditory cortex and regions like triangularis as hubs that sort of where the altered connections originate, and mostly, as I mentioned, go into the other hemisphere. And this is actually very intriguingly consistent with sort of recent increase of interest to the functional localization of language functioning and the optimality of the development of functional networks as potential players in neurodevelopmental disorders, and in particular, atypical lateralization, for example, has been recently implicated or shown in individuals with a history of developmental language disorder. But of course, given the sample size, this is intriguing, but relatively sensitive. We also found that, and so I'm not going to talk a lot about copy number variants in this population at all, in part because despite us finding evidence of association for several copy number variant regions in this population and the multivariate developmental language disorder phenotype, real-time quantitative PCR actually did not substantiate the presence of these copy number variants, which is problematic, but sort of, I wouldn't say expected, but it sometimes happens to CNV data obtained using intensity values from traditional SNP microarray panels. And this actually didn't really matter sort of what sort of processing framework or processing pipeline was applied, because we used pretty much everything under the sun to look at that, and we either didn't find anything or it didn't validate. However, we did find a significant association with developmental language disorder status in AS, and the cumulative burden of, or cumulative length of runs of homozygosity in several length bands, right? So 250 to 500 kilobases and 1 to 1.7 megabases. In these two ROH length brackets, affected individuals in the population seem to have longer cumulative lengths of runs, although we did not find sort of individual runs of homozygosity that would ideally drive these association with increased ROH burden. There is sort of another host of tentative findings for these runs of homozygosity, but nothing as pronounced as our SETBP1 finding. So DLD, or developmental language disorder, is actually a as I mentioned, is a multivariate and multifactorial complex disorder. It's just as complex and just as multivariate in the presence of sort of reduced background heterogeneity, for example, as it is the case for our population. And even here, developmental language disorder seems to be regulated by multiple genetic pathways, genes, and seemingly even different types of variants. However, we're able to identify SETBP1 as a novel candidate gene for developmental language disorder in this population, and a follow-up study using neural endophenotypes and network connectomics approach sort of corroborated these findings by implicating SETBP1 in the functioning of intercortical language networks during word processing in a mixed sample of affected and unaffected children in AS. And saying this is all I have. Oh, great, 40 minutes. And I would like to, of course, thank Golden Helix and also our lab, members of whom you can see right here, and several other collaborators that are in Norway, Wayne State University, and UConn, who contributed to these studies, as well as our funders, Northern State Medical Academy colleagues, and children and families who participated in this research. God knows it was burdensome. Thank you.
Thank you, Sergey. We will go ahead and take questions. I did forget to mention that at the beginning. Please feel free to enter them in the questions panes. I will have the recording and slides up on our webcast today or tomorrow, and I'll email them directly to those. So directly to you, excuse me, so look for them in your inbox. Our next webcast, we should be sending out email invitations fairly soon, so look for those. And let me look to see if I have any questions. My apologies again for not mentioning at the beginning. I don't see any at this time, so we can go ahead and wrap for today. Thank you everyone for your attendance, and Sergey, thanks for a great webcast.
Thank you very much. Now I'll be happy to answer questions if there are any by email or whatever. So thank you again.
Exactly, and feel free to email questions to info@goldenhelix.com as well, and I can email them to Sergey. Thanks so much, everyone. Have a great day.
Awesome. Bye.